Hello and welcome to my channel, Biology Made Easy for Exams and Interviews. Today we are going to talk about the topic called epigenetics. What is epigenetics? The word epi means above, something above genetics. That means it means some genetic regulation, some regulation of genes. So regulation of gene expression, which involves some mechanisms but not DNA mutation. Okay, so in bracket I can write not mutations, no changes in DNA sequences. Okay, but mechanisms like DNA methylation, then you can say histone modifications, and there are other mechanisms like regulations by non-coding RNAs like microRNAs, etc. But today we are mostly be focusing on these two mechanisms, DNA methylation and histone modifications. Now, epigenetics, as I said, is a change in gene regulation which is heritable. Heritable means if the cell divides into two daughter cells, the epigenetic mechanisms will be transferred from the mother cells into the daughter cells, or during the formation of gametes, these are transferred from the parent to the through gametes to the offsprings. Okay, so these changes are very important. And why we are interested, why we are studying this topic, because there are many diseases, including cancer and others, where epigenetic perturbation means changes or abnormal changes are very common. And the interesting thing is these changes can be cured or backed to normal by some epigenetic drugs. So there are some drugs which can actually cure or make this abnormal changes back to normal. So there is a hope that some diseases can be cured if there are these are caused by epigenetic changes or so. Now to understand this, we first need to understand, suppose this is a segment of the DNA. So DNA is double stranded and the strands are antiparallel in nature. So suppose this. Okay, so a segment of the DNA, it is called what? A segment of DNA is called gene. So suppose this is a gene. Now the gene starts from a region which is called the transcription start site. So I would say this is TSS. T means transcription start site. Now here, what is important to understand, the upstream of it. Upstream means this particular region is called upstream, and this is downstream of the gene. So the upstream region of a particular gene usually have a region called promoter. Now the promoter can be just adjacent to the gene. It could be far upstream of the gene also. It is called super promoter. So to make the topic simple, I will consider this particular region as the promoter of the gene. What is the function of a promoter? The function of a promoter is to recruit some factors called transcription factors, right? Transcription factors that will help to recruit. There are many proteins involved, to recruit the RNA polymerase. And what is the function of RNA polymerase? This will actually polymerize the synthesis of mRNA by the process called transcription, right? We know that. So this is the process of transcription, and we now get mRNA. Now mRNA will be translated to protein. We are not writing that now. So if this region, the promoter, have some DNA modifications like methylation. Okay, what is methylation? It's the addition of a methyl group, CH3. So CH3 is added to the cytosine residue. So we know that A, T, G, C are the four bases, right? So the cytosine, which is adjacent to guanine, So these are connected by phosphodiester bonds. If we study the DNA structure, you will remember. So this is the phosphodiester bond. So this particular methyl group, which is attached with cytosine, and that cytosine is adjacent to guanine, it's called CpG. Now if this stretch has methylation, so if I add some methylation, so dots, so make it dots. So if I have some dots here, so the cytosine is methylated, then this particular transcription factor will no longer bind, and as a result the RNA polymerase will not get recruited, and as such there will be no transcription or no mRNA production. So the gene is, we can say it is switched off, right? So if there is DNA methylation in the promoter, this is the gene promoter, then the gene is usually off. So this mechanism of silencing of genes, so it is called gene silencing, turning the gene off through a mechanism of DNA methylation, is a mechanism that falls under epigenetics, epigenetic regulation of genes. Now who does that? Who adds the methyl group? So the methyl group is added to cytosine, which is usually adjacent to guanine. Here methyl group is added by enzymes, enzymes called DNA methyl transferases, DMNTs, DNA methyl transferases. So you can understand that in some kind of diseases, this DNA methyl transferases are basically upregulated, and as such there is random DNA methylation along the DNA. Okay, so the opposite thing is also occurring within the cell. As I said, these processes are all reversible in nature. That means if an enzyme is adding methyl group, there are other enzymes that can remove the methyl group, removes the methyl group. It's called TET, T-E-T. So these enzymes, they can remove the methyl group. Okay, so summarizing what I've said so far, what is epigenetics? Epigenetics means something above genetics, genetic regulations that do not involve mutation but involve mechanisms like DNA methylation that basically silences the gene. Most likely they will shut down the mRNA expression. Then there are some other modifications called histone modifications. Now here I'm going to talk about that. So what are histones? Histones are basically proteins that binds with the DNA or that wraps around DNA. Now you know that this there are how many histones? We know that H2A, H2B, H3, H4. There are 4 types of histones that that into 2. So each of these histones forms an octamer. Octamer means 8. So 2, 4, 6, 8. 8 histones, they form a nucleosome. So if you see like this, 1, 2, 3, 4, 5, 6, 7, 8, it forms a nucleosome that around which a DNA wraps. Okay, and these are the units. And then between 2 adjacent nucleosomes there are a gap of about 150 bases or 149 bases, and there is a linker histone which is H1. So there are 5 types of histones: H2A, 2B, 3 and 4. They form the core nucleosome, and there is another histone H1 which forms a linker histone. Now this particular histones, all of these histones can be post-translationally modified. So why? So this is a new term, post-translation modifications. Okay, modifications. That means after translation, after the protein has been formed. So as I said, histones are nothing but proteins. These proteins, after they are translated, these can be post-translationally modified. Now, a key component of these histone proteins, component means the amino acid of these proteins are lysines. Lysines are positively charged nature. So these histones are basically positively charged, plus. Okay, and the DNA is, we know that DNA has the phosphate backbone that gives its negative charge. That's why this positive charge and negative charge of DNA and positive charge of histone, they attract one another. Okay, they actually attract one another and they form a tight complex. As you see here, this nucleosome is formed because these are two opposite charges. So these DNA can form, or they can tightly wrap around the histones. Now, this positive charge of the histones is contributed by lysine residues. So if these lysine residues are post-translationally modified, for example, they can be methylated. I would first start with acetylation. If they are acetylated, then they become neutral. They become neutral by charge. And then this interaction between the DNA and the histones falls apart. So earlier the DNA, say, was wrapped around histones. Suppose there are histones inside. Now, if these histones are acetylated, now what are these threads that I'm drawing? These threads are nothing but the 3' tails. So this is super simple structure. This is a histone, but this is not a circle protein. This is made up of amino acids. So these amino acids are wrapped around. That's why it is giving like a shape like this. So the amino acids are having N terminus and C terminus. That I will tell you when I will talk about translation. But for the timing, take my words. These C-terminal proteins or C-terminal end, they have lysine residues, and those are acetylated. So once these are acetylated, then the DNA does not bind. So the DNA structure falls apart. Okay, the DNA becomes open. So if, think about this, if you see that there is a transcription start site here, then this transcription start site becomes free to bind to different kinds of proteins. For example, here the proteins cannot bind. Here the RNA polymerase transcription factor can bind, and from here, if this is the gene, the mRNA will be produced. Okay, here is no transcription. No transcription because there is no place for the transcription factors to bind. This is wrapped around the nucleosome. Okay, so what is the fate? So if the histones are acetylated, then the promoter, the gene promoter, is free. Then what happens? The transcription factors can bind, RNA polymerase can bind, and then the gene is on. Okay, so again coming back, so if there is DNA methylation, then there is gene silencing. If there is histone modification, if there is histone acetylation, then it is activation, or gene is on. Okay, more expression. Now again, what factors or what enzymes are involved? So the addition of, say, the histone, say H3 to H3 acetylation, okay, this is actually done by some enzymes. So examples like P300. The name of the protein is like that. It's because of its molecular weight. CBP. Okay, PCAF. Like this. And again, just like the DNA methylation process, this is reversible in nature. That means the acetylation can be deacetylated. This is done by histone deacetylases, HDACs. And there are how many HDACs? 1 to 11. There are 11 types of HDACs. Okay. Now, what residues within the histones get acetylated or other modifications, that is very specific. For example, in H3, lysine is K, right? Lysine 9 can get acetylation, H3K14 can get acetylation, H3K27 can get acetylation, like this. There could be more, H3K56 and etc. So these are actually activation marks. Here, if the genes are acetylated, or if the promoter of a particular gene has this modifications, that means the gene expression will be higher. Okay. Now, if I talk about other histone modifications, then that thing is the histone methylation. Now, histone methylation. Now, you may also note that we talk about histone H3 methylation, acetylation, H3K9 acetylation, or H4 acetylation. But why not H2A, H2B? H2A and H2B, these are core histones. Modifications of the core histones do not rupture the nucleosome structure so easily, but or rather you can say that the commonly H3 and H4 histones, they are commonly more acetylated than H2A, H2B. The other histone modifications are histone methylation. That is something complicated. Now, for example, H3K9 methylation is a marker of gene suppression, but H3K4 trimethylation is a marker of gene activation. So for methylation, histone methylation, it can mean both suppression as well as activation, but for histone acetylation, it is generally, almost universally, all these acetylation marks are related with activation. Okay. Now you may ask why this particular methylation is a suppression marker. It is suppression and marker not just because of this mechanism of wrapping or open conformation of closed or open conformation of nucleosome. There are other factors, because this kind of modifications recruit some repressors that silences the gene, that switch off the gene, not just physical conformation is involved. So that is a little bit complicated process. So summarizing what I've said so far, so epigenetic regulation is a gene regulation which does not involve any mutation but involve this kind of mechanism. DNA methylation means addition of a methyl group to the cytosine residues which is adjacent to guanine. Histone modification means addition of acetylation mark or methylation mark with histones like H3, H4, which form the core histones, so that if any changes, these entire nucleosome structure will fall apart. DNA methylation is generally associated with gene silencing, because if this methylation is happening on the gene promoter, then it will prevent the transcription factor from binding, prevent RNA polymerase recruitment, and as a result the gene will be switched off. Whereas histone modification leads to what? Leads to open nucleosome conformation, from closed to open. That means if they are acetylated, then this the negative charge of histones is neutralized and the DNA cannot interact. The DNA is negatively charged, the histones are positively charged. So if this neutralized, this interaction falls apart, and then this promoter is now available for binding various transcription factors. Okay, so in general, histone modifications by acetylation leads to more gene expression. The enzymes that do DNA methylations are, here you are, the histone the DNA methyl transferases, and the reverse is done by 10-11 translocases or TET enzymes. Here the histone modifications are done by enzymes called histone acetyl transferases, which does, for example, these are the proteins, and opposite is done by histone deacetylases. There are 11 HDAX molecules. Okay, so in the next video I'm going to talk about, there are many techniques by which you can detect whether a particular segment has DNA modifications or histone modifications or not. Thank you very much for listening to the video. If you find this useful, please help by subscribing this channel and liking this video. Thank you.